DDNTP is a chain terminator. It stops that fragment from being built beyond that point. So it's going to stop whenever you add in DDNTPs. That seems a little disruptive, right? You're going to be adding, going along, and then if you add a DDNTP, it stops. So the idea behind this is to create a whole bunch of fragments that all stop at different positions. Now I've created, let me see if I can find these notes. At one point when I was teaching online, I made this series of notes that actually I think is helpful. And I put them in here. So let me use this to kind of explain Sanger sequencing notes. Okay, so this is what you're putting in the reaction, your template, one primer. You're putting in DNTPs, all four. And then, this is an extra note that I didn't have on that other slide. You put in DDNTPs and they are labeled. They're actually fluorescently labeled. So they all have different colors. So if you add a DDATP, for example, it's going to give a green color that can be detected by a machine, etc. So each one of these has a different color. Then you have your heat stable polymerase. You actually also use the same machine. That machine in the back can do sequencing. So you use a thermocycler machine for the sequencing reaction. So first step for doing sequencing is, again, to heat. So what temperature do you think we use for this step? 95 degrees. It's very similar to PCR. So we're going to heat it up. Your template is going to denature. Double-stranded DNA goes to single-stranded DNA. Then we're going to cool just like we did for PCR. It's the same step. It's an annealing temperature. So the primer, but this time only one primer is going to bind. And so typically, yeah, that temperature is around 55. Then we raise the temperature just like we did for PCR, and it's going to start adding. Now, mostly what we added in the tube was a lot of DNTPs. The DDNTPs are in a much lower percentage, so it's a much more often we're going to randomly add a DNTP to extend, but sometimes by chance we pull out of the hat, we pull out a DDNTP instead of a DNTP. Whenever that happens, that stops it. So wherever it is in the chain, that's as far as it's going to go. So you can see by chance this happens at all different positions. And you get different lengths of fragments. So when we're talking about PCR, what was our product at the end of the PCR reaction? Yeah.
Amplification of the DNA.
Yeah, one single fragment, right? It was all the same uniform size. You can see that the product for sequencing is different. You're getting many different sized fragments. The goal is actually to get fragments that end at every single nucleotide. You want to have a difference of a single nucleotide like this, so that you could check to see what was added at the end of each of these fragments. And when you check to see what was added, you actually are reading off the sequence. So after you've done this whole reaction about 30 times, you hopefully have a collection of different fragments. Then you need to resolve them. And in the olden days, they used to use big sequencing gels, really long sequencing gels. Nowadays, and we actually have this machine downstairs in the S110, it's a DNA sequencer. We use capillary electrophoresis. So there's a tiny tube, and the fragments are resolved through a matrix. Which one do you think comes off first? Which one's going to go through the matrix the fastest? The biggest or the smallest? Smallest. So the very first fragment to come off would be this red one. That comes off first. Then by size, they're going to come off one by one. So you can see red, blue, blue, orange, red, then it would be green and orange. Every single time something comes off, you have a detector that excites to basically collect the image of what color is coming off, and you get this. Anybody recognize this thing? You saw that, right? This is the chromatogram. That was what the computer was detecting. And it makes a base call based on what color it could detect. So you can put these letters, assign them according to which fluorophore matched up with what. Usually for sequencing, there's about a limit of like 700. So you really only get about 700 nucleotides of good sequence. What happened at the beginning of your sequence and the end? Does anybody remember looking at those chromatograms?
They were like really difficult.
Yeah, we did not have, non-overlapping peaks. There was like multiple peaks, and that's common at the beginning and the end of the run. So really for this, it's a limit of 700 nucleotides at a time. So this was the method that was used for the human genome, and this is how long 3 times 10 to the 9th for a halfway genome base pairs. We're doing 10 to the 9th, you have to think about how big that is. 700 nucleotides at a time. That's going to take forever to put all those pieces, right? So this, like I said, this is still a very useful technique. In fact, we could do it, we have the kit to do sequencing in our lab. We could do it ourselves, but it's really limited to short things. It's more useful for short pieces of DNA compared to like whole genomes. Anybody have questions about the process for this? These notes are in here, so if you want to come back and refer to those, you can. Okay? And this kind of just shows the same concept, right? You're generating fragments, you resolve them through that capillary tube, and then you take pictures of it and you get the chromatogram. All right, so for the human genome, I do want to show this. I don't know if I have time to show this video. I had shared that this took 13 years. The first draft was published actually in both Nature and Science. Those are the two biggest, that and Cell, those are the biggest science journals. In 2001, they published the draft. That was one person's genome, right? And obviously we know that there's a lot of variability, or there is variability, so this was not representative of all humans, but it was a good reference sequence now that we base a lot of stuff off of. The technique that was used for this, I want to talk about two different approaches, because actually there was one group that did this, and then there was a company called Celera that did it at the same time. And they actually ended up collaborating and sharing their data, which made it possible for that sequence to be published. But the technique that was used was called shotgun sequencing. And there was two different approaches. So one approach, this top approach, this is just pure shotgun sequencing, where you take all of the genome and you just break it up. It randomly gets broken up into these smaller pieces, and you sequence those individual pieces. So I want you to think about, just to give you the context of that, that would be like, anybody do jigsaw puzzles? Like little 10-piece jigsaw puzzles? Just to consider how big this is, this would be like if we had a jigsaw puzzle the size of this entire floor, probably, and you broke it up and each piece is about the size of this jigsaw. And to reassemble it, you have to figure out where this piece goes in this entire S building floor, right? So that's how they mapped. Once they figured out the sequences, they had to put them back in order. And that was called ordering the contigs. These pieces that they have that are sequenced have to be reassembled. So that, as you might imagine, would be very, very time consuming to figure out, especially when you consider that the human genome has a lot of repetitive sequences, right? Because that's going to be confusing if there's repeats. How do you know where this particular piece goes? The other company used this approach. This is called hierarchical. They decided, you know what, let's keep things like in sections. So they broke it up into sections first and put these in big vectors, like bacterial artificial chromosome vectors. And then they would say, all right, from this piece, now we're going to break it up, but we know when we reorder it, it has to go back into this section. So that would be like, if my jigsaw puzzle right here, I say, all right, well, I'm just going to break up this right here, and then I reassemble this, I know where this table is in the layout of the whole building. But this is really time consuming to do this first this first step. So together, because they shared their data and they made it available, this group could use some of the work that this group had done and vice versa. So they ended up, it actually worked out because they ended up being able to share what they had found to be able to put those sequences together. So that's the two different techniques. Nowadays, as I said, we're not, for genome sequencing, we're not really using Sanger sequencing anymore. This is one of the ways that we can do next-gen sequencing. And there's actually multiple different ways. I was going to show you this short video. You do not need to know how they do this, but it's fascinating that they can do this. So we'll just watch this very short YouTube. And also, amazingly enough, BC, I don't know how it is, BC always has these equipment grants, or the state will say we have leftover money, do you want to buy anything? So we actually have an Illumina sequencer downstairs. Nobody really uses it because it's, you have to be trained on how to use it. Anyway, but we have one. So if you ever wanted to see what the Illumina sequencer looks like, we can go down there. Okay. Put this video in.
Sample preparation begins with extracted and purified DNA. The first step in Nextera sample preparation is tagmentation. During tagmentation, transposons simultaneously fragment and tag the input DNA with adapters. Once the adapters have been ligated, reduced cycle amplification adds additional motifs, such as the sequencing primer binding sites, indices, and regions that are complementary to the flow cell oligos. Clustering is a process wherein each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over, and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The three primins are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, 4 fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determine the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template.
Okay, so they go into a lot of detail, which we don't need to watch, but if you kind of saw the idea is massive parallel sequencing, so instead of just doing one plasmid with one sequence, it's all being done at the exact same time on a glass slide. And so this is really, I think it's amazing this, they can even mix multiple people's genomes onto the same glass slide because they have barcodes. So now they're talking about the index. So they can use different barcodes to say, to kind of map each of these sequence reads to one person or another person. And as you saw, that glass slide is, every time a new nucleotide is added, they take a picture. It looks like Starry Night. If you actually go into a sequencing facility when they're collecting this, it's just flashes of color changing, and they're collecting all that information. And then they have to, once they have the information, then they have to map it back to the reference. It's now good that we have a reference so you can take a stretch and say, okay, this belongs to chromosome 20 or whatever. So that's why things are so much faster. That's why you can do it in a week compared to 10 years that it took in the beginning. So anybody have questions about that? Anyway, I think it's phenomenal that they have come up with, that it has, just progressed to that level. So, okay, so I think we've kind of covered, so don't worry about the details of this, but again, I introduced it just so that you could share with your family members or whatever about, how they're able to do sequencing so fast now. The last couple slides before we will do our lab, and then we'll come back and talk a little bit more about biotech. But one of the things, once they did this, I think people were really shocked because they expected to find a lot of the human genome actually being genes. First of all, humans are so complex compared to all these other organisms. They were shocked because the percentage of actual protein coding exons is less than 2%. So that means that most of our genome is not making proteins. And that's phenomenal because why would we maintain all of this DNA if that's not its purpose? That, the paradigm has always been like DNA is used to make RNA, which is used to make protein. But so there's a lot more for us to learn, like what is the purpose of all of this other stuff that's in there. So you can see about a quarter of the genome is the introns. So, of course we talked about alternative splicing as being a benefit to having introns. Half of the genome is transposable elements. Anybody know what those are? Transposons or transposable elements? May not have, I don't know if people talk about that in like Bio 160, but transposable elements are jumping, or I guess we could say mobile DNA, not jumping. But it technically can, if it's active, it can move around. So transposable elements are mobile DNA elements, meaning that they can move around. Some of the ones that are still in the human genome are no longer active, so they're no longer moving around. But people are still trying to understand, this is a huge burden every time you replicate, so why is it being maintained? What is the selective advantage to have that, right? Because we're inputting energy every time we replicate, and we continue to maintain those. A lot of the other stuff is pieces that we've discussed. So we've talked about centromeres and telomeres, repetitive sequences that are part of the chromosome. There's a lot of sections that are repeats. So there's short sequence repeats like micro and mini satellites. And then there's a lot of this stuff that's non-coding, and people call it the dark genome, and they don't really understand what its purpose is. So there's still research going on to figure out why do we maintain this? If, what is its exact purpose? So I guess I'll skip that slide. Once we have this information, what have we done with it, right? So now that we have whole genome sequence, we have reference sequences, how many of you have actually sent off DNA of your own to be looked at like 23andMe or Ancestry.com? So there's sequencing services. They actually are not giving you a whole genome sequence. What they look at instead is these SNPs, single nucleotide polymorphisms. And those are places in the genome where there is a high level of variation. So for SNPs to occur, they have to be higher than 1%. If you had a change to your sequence it could be from a mutation. If that just happened in a small fraction of individuals, that would be a mutation. But there are places where there's this variation that they've noticed, and they have associated certain SNPs with certain traits, or with even health traits, right? So if you have a particular SNP, it could mean that you have an increased risk of some disease like Alzheimer's or diabetes or other things. So they can be associated with different diseases. If you send off for 23andMe, they give you an option. Actually, it costs more if you want the health report. So if you want to get the SNPs and the information on your health alleles, you have to pay more. And they would say, like, you have this percentage chance of developing whatever. I personally couldn't do that. So even though I love genetics, my husband and I did our 23andMe, but we just got back to the ancestry stuff, and I thought about it. He's like, well, do you want to do the health report? I just can't. I'm too much of a worrier, and I feel like if I know that I have some allele, I will just spend all my time just worrying about, okay, I'm gonna get this disease or, anyway, so I would rather not know. Some people want to know because maybe they would take extra steps to be precautious, but I personally, I don't want to have the knowledge that if I have some increased risk, because a lot of times the risk is like, it's a small difference, like you have a slightly increase, maybe it's like 6% instead of 3%, but still, I would just spend my time just worrying about it. So anyway, the other thing that we can use SNPs for is actually mapping. So like with different diseases, a lot of times the goal is to figure out what is the causative gene, which genes are involved in it. So like, for example, autism. We don't really understand what causes autism, right? If we knew which genes or pathways were involved, we might be able to look at treatment, or how could we kind of modulate this pathway. So these type of studies have been used, SNPs have been used to identify what are areas of the genome that always seem to be a certain sequence for a certain individual with the disease. So they do these genome-wide association studies. They're called GWASs, and they find places in the genome that, maybe this population with autism might have a certain sequence. The other thing you can do is track change over time. This is why they can, at 23andMe, they can tell you what your ancestry is. The closer your sequences are, that means that you're more evolutionarily closely related, and that's how they have assembled these different kind of ancestry types. All right. So yeah, with this, when you send off for like 23andMe, they're using one of the 10 million SNPs in the genome to classify you and give you information. All right, so let's take a little break. I think that's kind of the end of the sequencing section. So maybe like a, 7-minute break. So let's come back at 1:15, and we'll start these overnight cultures, and then with the very end of class, we can talk about like gene therapy and CRISPR. Those are the two main topics I want to make sure that we get.
I saw some